a sophomore in the Randall Research Scholars Program. I work in Dr. Guy Becker's lab in the Department of Biological Sciences here at UA, and today I'll be presenting on our project analyzing the effects of climate change on the composition of amphibian microbiomes. After introducing my project, the Becker Lab, and the importance of our research, I will then provide some relevant background information on amphibian microbiomes and the fungal pathogen BD. From there, I will discuss the experimental design of our project and ongoing data analysis, and finally I will finish with future research directions. As climate trends continue to change, we are observing massive die-offs in amphibian populations, specifically anurans, which include frogs and toads, all across the world, resulting in disastrous ecological imbalances. These die-offs can be attributed to the emergence of a fungal pathogen known as Batrachochytrium dendrobatidis, which I'll be referring to as BD over the course of this presentation. The Becker Lab is interested in the interactions between an amphibian's skin microbiome and the fungal pathogen BD. Accordingly, our project investigated the effects that simulated drought conditions have on the composition of an amphibian's microbiome and its interaction with BD. So the skin microbiome is of great interest to those studying BD because BD is transmitted through the skin of amphibians. As you can see, zoospores travel through water where they will ultimately implant themselves into the skin of an amphibian and develop into sporangia. These sporangia disrupt processes like osmoregulation and actually inhibit gas exchange. This often results in the death of the amphibian. So what is the microbiome? In the context of this presentation, the microbiome is all of the bacteria that reside on the surface of the amphibian's skin. As I previously mentioned, BD is transmitted through the skin of amphibians, and thus microbiomes that contain bacteria that display antifungal properties can actually mount two kinds of immune responses. The first of these responses is the innate immunity. Innate immunity functions as a barrier that prevents the initial implantation of the BD pathogen. The second kind of immunity is adaptive immunity. This occurs after a successful invasion of the BD pathogen. Here I have modeled a microbiome with each of the different colored circles representing a different species of bacteria, and as you'll see, the red skull indicates the invading BD pathogen. When BD invades the microbiome, it outcompetes bacteria that don't display any antifungal properties, and as it continues to grow and reproduce, it will actually further push these bacteria out of the microbiome. However, if there are bacteria that display antifungal properties such as the Jansenobacterium, these bacteria can actually flourish themselves and in the process push the invading pathogen out. This will continue until the invading pathogen has been completely expelled from the microbiome. Adaptive immunity is termed as such because the composition of the microbiome is retained after the pathogen has been fully expelled. The abundance of these antifungal microbes is retained, thus allowing a much quicker and more efficient response were the animal to encounter the BD pathogen once again. Furthermore, the composition of the microbiome is of great importance to this immune response because without bacteria that display these antifungal properties, no response is possible. Furthermore, innate and adaptive immunity usually occur across two separate mechanisms, but as you can see, the microbiome displays both, blurring the distinction between the two. To test the impact of climate change on the diversity of an amphibian's microbiome, we simulated drought conditions on two species of salamander that we collected here in the wild in Alabama. We also collected a species of newt to act as our BD reservoir, as they are natural carriers of BD. At the beginning of the experiment, we took two samples. The first to sequence the microbiome of each of the individuals, and the second to quantify the fungal load of BD in each of our specimens. Here you can see one of 6 of our replicate sites. The salamanders and newts were placed in each of the smaller tents, and then one of the 2 smaller tents was then placed in a larger greenhouse enclosure. The greenhouse enclosure prevented the salamanders and newts inside from experiencing any external rainfall, thus simulating our drought condition. The other smaller tent was then placed outside of the greenhouse enclosure, and the salamanders and newts inside experienced natural weather conditions. Inside each of the smaller tents, we placed 4 plywood coverboards and constructed a water pool at one end of the enclosure. Every salamander in a given tent was placed besides the coverboard furthest from the water hole, and the BD-ridden newts were all placed inside the water hole. Furthermore, the coverboard closest to the water hole actually overlapped the water slightly. This allowed salamanders that traveled from one end of the enclosure to the water hole to possibly encounter BD from the newts. We suspected that the drought conditions would lead to salamanders aggregating around the water source and thus experiencing greater levels of BD transmission. Our experiment ran for about a month, and at the end we sampled both the microbiome and fungal load again. Every measurement and sample data was then compiled into our metadata spreadsheet. We were then able to use the bioinformatics software QIIME2 to process this metadata spreadsheet into OTU tables and taxonomic plots that we could visualize. OTU stands for Operational Taxonomic Unit and simply describes how we classify these bacterial sequences by their phylogeny. We were able to do this without knowing the species of each bacterial sequence. Here you can see one of the taxonomic plots that we were able to visualize. It contains 13 OTUs out of the almost 17,000 unique bacterial sequences that we identified. These 13 OTUs also occurred across the largest number of samples. As you can see, the 5th and 10th most common OTU are actually the Jantanobacterium that I mentioned earlier displays antifungal properties. Displayed here, I have the exact number of sequence reads of the Jantanobacterium from a group of specimens that I've displayed here. You can see that every specimen has Jantanobacterium reads. So QIIME2 allowed us to visualize the abundance of the Jantanobacterium in the specimen using these OTU tables and visualization plots. From here, we are able to move to JUMP, which is a statistical analysis software, which allows us to investigate the statistical significance that any of the experimental variables may have had on the Jantanobacterium in the microbiome. So we are still currently working on finishing the data analysis for our project, but we do have some preliminary results indicating that there is a statistically significant difference in the mean number of reads of Jantanobacterium across the two treatments. As you can see, the mean number of reads of Jantanobacterium across both species of salamander was less in the group subjected to drought conditions compared to the group that was subjected to the natural weather conditions. This indicates that the simulated climate change had a negative impact on the climate change of the microbiome and thus possibly affecting the organism's response to a potential BD infection. This brings us to the current stage in our project. Looking ahead, we'll be finishing the data analysis for this project, and next semester a graduate student in our lab is actually looking at inoculating salamander species with bacteria that display known antifungal properties. They will then investigate the effect that this has on the composition of the amphibian's microbiome and its interaction with the BD fungus. Furthermore, continued research into the effects that climate change and other anthropogenic disturbances like habitat fragmentation and deforestation have on the composition of an amphibian's microbiome is necessary. And finally, research into the effects that human microbiomes have on disease mitigation are very relevant right now. Before I take any questions, I'd like to give a special thanks to Dr. Greenspan for teaching me all the bioinformatics this semester, and of course Dr. Becker for allowing me to work in his lab over the course of this project. I would also like to mention Alabama Audubon, the NSF, and Alabama Water Institute for the funding they have provided for this project. Thank you for your time. I will now take any questions.
It's clear that BD is a very destructive pathogen. So did you wipe out all the salamanders that were used in the experiment, or what did you do with the salamanders at the conclusion of the experiment?
Great question. So BD actually doesn't affect salamanders like it does frogs and toads. There's another chytrid fungus termed B. sal that is actually much more devastating to the salamanders. So the salamanders weren't that affected by the BD, and we didn't have a large number of individuals that tested positive for BD over the course of the experiment. So no, we did not kill off an entire population of salamanders in Alabama.
What are the impacts of the amphibian die-offs that you mentioned earlier?
Okay, so any large die-off, especially these extinction events that we're seeing with these amphibians, have huge ecological impacts in that they affect food chains, food webs, predator and prey interactions. But one that particularly pertains to humans is the increase in disease from mosquitoes. So contrary to maybe popular belief, frogs aren't very big predators of mosquitoes, but tadpoles actually consume similar food that mosquito larvae do. So if frog populations decrease, the number of tadpoles will decrease, and as a result, the mosquito larvae aren't competing with as many other animals. So their numbers will increase, and as a result, mature mosquitoes will increase. And mosquitoes are the most devastating disease vector known to man. So there was actually a paper published recently that tied BD to increase malaria transmission in Costa Rica.
So with these extinction events, what are some possible steps that can be taken to prevent them?
So there are antibiotics that are available to treat BD in these amphibians, but the challenge with that is mass deploying these, and there's not really a feasible way to do this. So really the goal of this research and similar projects is to bring attention to the effects of climate change so that we can bring attention to the issue and hopefully have legislation passed to address the main issue, which is climate change. None of this will stop if we continue to see these changes in climate trends, so we need to address the root of the issue, and that will be done through the passing of legislation.